starting our class. But if you're just getting here, you can collect those, or you'll have time to collect the building supplies when we get to our in-class assignment today. All right, so here we are, and I'll talk about our exam. I almost finished grading them. I didn't quite finish grading them, so I'm not going to hand it back today. I apologize about that. And we'll talk about it in a minute. This is looking ahead, and so looking ahead to our second exam. So we're going to be heading to our second exam, and it's going to be just before Thanksgiving break. And we know what it's going to cover. There's a typo in the syllabus. I left off chapter 13. Oops, but it's going to cover 4 chapters. We'll talk about fruit flies today, and chapter 2, and it's just a gigantic chapter. And it's chapter 2 in our book. We skipped over it because it's just such a monster chapter. Then we're going to jump to chapter 13. It's our only chapter in our textbook that deals with plants, and it's going to be really a compare and contrast plants and animals. And that'll be like a big question on the exam. Like a 20-point question will be to describe the comparison of the similarities and differences between plants and animals on Earth. Because the last common ancestor between plants and animals was a single-cell eukaryote. So after those diverged, plants invented multicellularity, and so they invented development, and animals also independently invented it. So there's no such thing as an orthologue in developmental mechanisms between plants and animals. There are orthologs of things like DNA polymerase, RNA polymerase, and proteins and ribosomes and so forth, because our common ancestor was a single-celled eukaryote that already had all those cellular functions. Then chapter 7, and chapter 7 is back to animal development and talking about general principles of animal development. And I sort of unofficially subtitle it the cell biology development. So we talk a lot about what you can see under a microscope, and we talk a lot about molecular mechanisms of gene expression and protein function. This is the cell. And so all living things on Earth are composed of cells. And so that's really the ultimate fundamental unit of development. And cell biology is a really satisfying topic for students. Actually, the work we're going to do today, the macaroni, is a big concept in cell biology. So we'll peek ahead a little bit at that, sort of foreshadow to that. And then chapter 8 is actually where the where the book deals with cell differentiation. So we started our course on that, and this is where the book talks about muscle differentiation. So this is going to be a chapter where we're like, oh, great, because it also is where it deals with stem cells, and we've already talked about stem cells a fair amount. And so it'll be a little bit like a review. It'll probably be the chapter in the book that's the most accessible to read, and not just be blown away by all the details the book has. And but we'll be expected to take it up a notch like that. And the second exam will be new material, but it just super definitely builds on stuff. So I bet you'll go back and watch the muscle differentiation videos again for the second exam, because it'll be a big part of the exam. So we're not going to be asking explicitly to go back and study from the first exam material, but to move on. But in moving on, we're building it up. Okay? So here's just a quick example of the cell biology of development. So again, we're going to do that topic today in fruit flies. So we're going to introduce this today, but here's an example from chapter 7 for the whole week. So here's some sponges, and they are the most distantly related animal on our planet. Are they even animals? Just barely. Sponges are diploblasts. Everything else is a triploblast. So very, very distantly related, 800 million years ago. So here's something that can be done with any sponge. It's got a very simple body, just cells on the inside and cells on the outside. It has one opening, it pulls some possible food in and tries to digest it, and then it pushes that food out like that, okay? So this is something you can do with a sponge in a lab. You just take it, send it through a mesh, and it will be separated into individual cells. And if you just leave the individual cells overnight, they'll reassemble to a sponge, a diploblast sponge. And the concept that we want to focus on is the reason they're doing that is because there are cell surface proteins on the surface of all the sponge cells, and they hold hands with each other. And there are some different types of hands, so they use that to organize their whole body. By just having the individual fundamental units of life, the cell, have specific hand-holding proteins on the surface and have a variety of them so that it can assemble different parts of its body. And those molecules are called cell adhesion molecules, and they were some of the original ideas in evolution of an animal body plan. And all animals have many, many cell adhesion molecules, and some parts of cells and tissues can actually reassemble like this. We couldn't reassemble a whole person, but we can assemble really some parts, and especially some of our early embryonic stories. Like if we separate ectoderm and endoderm, and then we just put those, mix them together in a dish, they'll separate out overnight because they have different cell adhesion molecules. So it's a real central concept, and that's the word cell adhesion molecules. And they're proteins, and so they must have genes, and the genes are in big families. We have big families of these cell adhesion molecules, and we'll learn about them in chapter 7. I'm showing this today just because the macaroni that we're going to use in our model assignment in class today is going to be a fruit fly story, but a big feature of it is the cell adhesion molecules. And we're going to have two populations of cells on our drawing, and so I brought in two types of delicious macaroni for two different types of hand-holding molecules, shell macaroni and elbow macaroni. All right, party. So very distant related, very ancient plan for building an animal body. Do plants have cell adhesion molecules? Nope. They might have some examples, but they're not orthologs, and they don't have that as a big feature. Just to mention something about that chapter, plants have cell walls, so they don't move around. Very different than animals, where the cells are going to jostle, right, to have the belt fold in, or the neural crest cells are going to migrate. Nothing like that happening in a plant. So really interesting to look at plants. So what should you do this week? So read chapter 2, and it's just a really big, crazy chapter. The book talks about 60 or so different fruit fly genes. We're not going to learn them all. We're going to learn a dozen. So a big part of this is, again, to build stories in class to navigate through the concepts that are found in chapter 2. Then we'll go over exam 1. I apologize, I didn't quite get it graded today. Obviously, I'll have it ready in class on Thursday. I grade the exams one question at a time, anonymously across the questions, like that. So I actually don't have anybody's fully graded quite yet. I'm about to start the last question, but the questions that are toward the end are the ones I leave that are the bigger, longer questions that are hard to grade. But I'll get that done, and there'll be this answer key video. And I'm thinking that I'll probably turn that into an assignment where it'll be you actually have to watch the answer key video and make some adjustments. Not to increase your exam score, but to have it as a separate assignment, just to get us back there, because the exam was really hard, right? People lost big chunks of points. Here's the deal. I know lots of you, I know you're superstars, and each one of you had really excellent answers on the exam. You'll end up doing really great in this course, but no matter how much I try to set it up, the first exam is just a brutal thing. We're like, gosh, what? We're really expected to know all those things. It was really hard, I agree. And so let me just show you this. It's back here. So because of that, so the first exam is just worth 20% of your final grade, and then the second is more and more, because I think the big point of college is to have different classes, different topics with different instructors, and become a flexible learner. So I really honor somebody that might be struggling to start, and they can increase their scores over the semester. So when I do final grades, I don't just crank them out mathematically. I do that, but then I also look at the scores and see how things are going, and we can talk about that. And when I hand back the exam, students will say, how can I do better? And I've been saying that the whole time. The key thing is what's happening in class. 99 of the 100 points on the exam were from class. they're complemented and strengthened with the textbook, but it starts in class. And that's a big reason why I record it. And you have access to that. If you weren't in class, so forth, like that, you know? Because in some days, some of the, things, there were like 10 points that were just from one class meeting. We talked about that in one class meeting, And if you miss that, my gosh, right? You're just, classmates' notes aren't good enough to have you, learn everything that was happening. So that's the big focus. And it's just to really figure out what's going on in class, and even stuff that we talk about briefly can just show up on the exam, okay? And in general, people did fine. Everybody's trying, everybody's gonna end up doing fine. I've taught this course a bunch of times, but it's true. The first exam is like, whoa, yep. So I'll get that back to you. We'll deal with that. And that's something we'll deal with this week as well, okay? And then the next thing we have to do is, it was on the syllabus just before the first exam, but I shifted the exam around and I missed that class, and so we still need to take the first step in our term paper, and our term paper will be due near the end of the semester. And the syllabus describes the term paper. It doesn't take very much to describe it. You don't have to look at it right now, but it's on our bright space, and that's where you should go. And what you do is the syllabus describes a specific journal, and it has a link. And you go to that journal and you pick one research article from that journal. So you're gonna look at something and see something like this. So there's the journal, it's called Development, and again, the link is in our syllabus. Then it has different sections, so it has these bold sections, and you're gonna pick one from the research articles section. Here's one about plants. So that would be a good one to pick because we're going to cover that, and so it's a good idea to just find something that we're dealing with in class and just kind of double dip. Then what you're going to do is you're just going to read that one research article. It's called a primary research article. There are new experiments, new data, new discoveries described in that research article, and the goal is for you to write a term paper like about 6 pages long, and that tries to connect that research with our course. That's the big thing that I'll look for as I read it. And so a big part of the term paper is the first 2 pages can be just you writing stuff you already know. Defining orthologs, defining model organism. Almost all these papers, they pick one species, a model organism. That's a little flowering plant, Arabidopsis, but it might be mice or fruit flies. They'll pick one species, and really often it's one or a couple of genes they're working on, and then it's a developmental topic, like formation of a neural tube. And pick it, and if you can, either print, if you have a printer, just print the first page, because we'll talk about those. I'll say who's got one, and I'll pick it up, and I'll just show you the strategy of how to tackle that, okay? Or have it on your device so you can pull it up and we can just look at it, and we'll look at a few of them in class a week from today. And then the next step to do after that is after class or sometime, just talk with me for about 10 minutes because I can just scan through it and help say, yeah, this is what that's about. And you can start getting a handle on how it fits in, like it might be about neurobiology, and that's one of the later chapters in our course, and we might not even cover it. So you can look for an article on a topic that you get a kick out of, so you can, of course, you can search like that. I think the most recent 6 months are not free. You should not pay for them. You can request those through the library, through interlibrary loan, or you can just go back more than 6 months and you can just download free research articles that are older than that. Don't go back several years. This field is changing really fast, so make it in the last few years, like that. Yep. And then talk to me about it. Don't pick another journal because their other journals are just not as good. So, so we want to get that going, and then where's our little list? So, chapter 2, go over exam 1 when you get it. Then find a primary research article. It'll take you half an hour, or at most. Then we'll do this assignment in class today. All right, and so next Tuesday, next Tuesday, so a week from today, bring the front page of that. either on your device or in a print, and so we can just say, Naomi, you got one? And you're like, yeah, here it is. And I'll say, oh yeah, look, this is about frogs, blah, 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 and help, get a feeling for, yeah, we can tackle it. And the big goal again is these are new discoveries, and can we use what we're learning in our introductory course to understand basically what's going on? You don't need to understand all the details. We'll talk about them more as we move forward in the assignment. The assignment is largely one you're going to do on your own, but we'll be talking about a little bit during class and definitely meet with you at least once and maybe multiple times. So in fruit flies, we're going to turn the fruit fly chapter into 3 clear stories. And you don't need to scramble to write this all down because we're going to talk about them one at a time. But it's going to be that entire chapter, and we're going to boil it down to just these 3 stories. And the stories are different than our bodies. Our first exam was all about chapter 3, 4, 5, and the introductory chapter, and that's all vertebrate. So these are going to be stories that are different than us, but they're going to feature some of these regulatory proteins, transcription factors, and signaling factors, and signal transduction pathways that are orthologs. So it's like this thing where we say, to understand car mechanics, we're going to turn to a motorcycle. And it's got different parts, but the tools used are common. You can learn about how the tools put the motorcycle together, and that'll help you understand how cars get put together. So that's the logic, and it's all down at this molecular level. We'll talk about cells, but the learning is all about these stories, and it's about how cells become different by patterning, by cell communication typically, sometimes a little bit of unequal cell division. And so we'll talk about Wnt, that's the Wnt signaling pathway. Your assignment that we're going to do in class, the center of it is just repeating that in a certain context in fruit flies. Yep, okay? And so here's the setup for it. So really, a lot of these genetic screens, we learned about nematode genetic screens, so fruit fly geneticists did the same kind of genetic screens. So induced mutations by exposure to a mutagen like X-rays in a lab of tens of thousands of little fruit flies, and then screening them, actually looking for a certain mutant phenotype, and then analyzing that mutant phenotype by complementation crossing and analyzing the genes and so forth. So here's just an example that's not covered in chapter 2, but it just provides you a context of understanding the work that we're going to be talking about. And the original work was done 30 to 40 years ago, where people did these genetic screens all over the world. These scientists were doing these genetic screens, finding these developmental genes. Okay, and so this is just an example. So here's a wild type fruit fly, and the scientists were, let's try to understand how the eye forms. It didn't exist, and it develops, it forms. So let's look for mutants in a big genetic screen that have no eyes, okay? So that's what they did. That was the mutant phenotype, and they found those genes. They did complementation crosses, what genes did we find, and then they analyzed the genes like that. So here's the name of one of the genes. And the reason that we're showing this example is simply this. When fruit fly geneticists describe and name a gene, they really often choose the mutant phenotype. And that seems contrary to your intuition. This gene they called eyeless. That's the name of a gene in fruit flies. And it's because the mutant that lacks a function of an eyeless gene, has a null allele of eyeless, has no eyes. So see, it's weird because eyeless is required to make eyes, but the gene is called eyeless. So we'll see that some, that they name things in an odd way. And in general, we've already mentioned, fruit fly geneticists are a bunch of goofy weirdos and name genes really bizarrely. But that's a common thing, okay? So just to mention eyeless is a famous gene that was studied 40 years ago, and it has a different name now because orthologs have been found in every animal on our planet that has eyes, I won't write it down, but it's called PAX6 now. Here's what the fruit fly geneticists found really fast. They found that if they took the wild type eyeless gene, so the one that's required to make eyes, and if they did an experiment where they forced the expression of eyeless in other parts of the fruit fly body, they would make wings with eyes or legs with eyes. And that's the same story as we saw with MyoD and the idea of the muscle precursors. The same thing is happening here, where this eyeless is like that MyoD. It's a transcription factor that's going to activate target genes that are required for eye development. So it's just this cascade, these dominoes falling, and it's the early steps to make eyes. And so it's just a fascinating gene because of that. And it's also fascinating because there's an ortholog on this in every animal that has eyes, but before this gene was discovered, evolutionary biologists had looked at the variety of eyes on our planet, which are very diverse. There are eyes on the edge of clams, so they can look at you with their shell closed, little tiny eyes. And octopuses have eyes, and lots of vertebrate eyes. Fruit fly eyes are profoundly different than ours. And evolutionary biologists had concluded that eyes had evolved independently 30 or 40 times on Earth. But then developmental biologists discovered that the molecular mechanisms regulating eye development are the same. So 30 years ago, that was a shocking discovery that is a big part of modern developmental biology. And that is, we see this tremendous diversity of life on Earth. But when we drop down to see how that life gets put together, there's tremendous commonality, shocking commonality, where you're like, what is that? How does that even mean that there would be these many different kinds of eyes that evolved independently, yet they share the same molecular mechanism to put them together? And it really shifts our perception and understanding of development in the context of evolution and the story and understanding of history of life on Earth. Okay? So here's just a little bit more, and this just underscores how wacky fruit fly geneticists are. So they isolated several strains, right? And then they did these complementation crosses. And every time they complemented, it's a typo. When they complemented, they must be a different gene, right? When they don't complement, it's the same gene. They're alleles of the same gene. So they found several different genes just through complementation crosses. And this is what they named the different genes. So they're like, we found a gene, we want to call it eyeless. Here's a separate gene that's required for eye development. What should we call it? And so let's call it eyes absent. And you're like, that's great. How about another gene? What could we call it? And somebody's like looking out a window and going, how about eye gone? Would that be a cool name for a gene? They're sure, why not? There's one, cineoculus, oculus, and it's like, that's Latin. What does it mean? And it means without eyes. They all are named based on that mutant phenotype, like that. And so, again, not the centerpiece. We're not going to see eyeless. You could bring it up as an example on a test. When I say, give me an example of some cool ortholog story, you could bring up eyeless. But it's not going to be a thing where it's fill in the blank and it's eyeless, because that's not it doesn't come up in our chapter. It just sets the stage for talking about fruit fly molecular genetics. And so our first story is going to be our assignment. And our first story is called Creating Compartments Within Each Segment. And these compartments are going to be cells that want to hold hands with other members of their compartment. So the story is very much like this. We have one class, but imagine we say we need 2 teams. And we're going to separate the class into 2 teams simply by having 2 groups of people holding hands. So you all get together and hold hands, and you all get together and hold hands. When you're holding hands, there's no mixture. There's a segregation. There's an invisible line between the 2 teams. And the line is called the compartment boundary. Okay? And it's based on those cell adhesion molecules that have been around for at least 800 million years, and animals use them in a bunch of different ways. We don't have the exact same story in us, but we use these molecules for that, and we have lots of different ways of building these compartments to organize different sets of cells. You know, we have one population of cells, and we need to start making things like a brain and a pancreas, so let's get populations organized. So that's going to be the story, and it fits into this. So here's a couple pictures of a caterpillar. There's a lobster tail, because this is a story not just of fruit flies, it's all arthropods. So all insects, spiders, ticks, mites, crabs, lobsters, all have a very rigidly segmented body plan. So you see that really well in a caterpillar, segment, or a lobster tail. Those are individual segments. And each segment in all those different animals has cells in it. And the cells are organized into two populations very early in development. And those two populations work as a team to define an invisible boundary called a compartment boundary. And that's sort of the keystone, the reference landmark for development to proceed. You might know something about when they're going to build a new building. They have some surveyors come out, and they measure and they establish some invisible lines to make the building level. So that's what's happening in development, is the two populations of cells say, I'm on team A and I'm on TP. Team A is the anterior team, team B is the posterior team, and we're just going to shout at each other and establish a line between our teams, and then all of development can be using that as a reference line to say, where are you in the reference line? So every leg on any arthropod has an anterior portion and a posterior portion, and there's no way to see the line. It's not represented in any visible way. There's a line that was set up very early. In fruit flies, it's set up just 2 or 3 hours after fertilization. They establish this line in each one of these segments. So it's the same story for each segment in the caterpillar. It's the same story for every segment in every arthropod. Very, very early in development, in the embryo, this line is established, and all development proceeds around it. Every insect that has a wing, there is made with an anterior compartment and a posterior compartment, and it develops. And in fruit flies, it starts about 2 or 3 hours after fertilization, and at that time, each segment is just 4 cells wide. So that's how early it is. The segments are eventually going to have, tens of thousands, hundreds of thousands of cells, but when it's just 4 cells wide, it says you're anterior, you're posterior. That's going to be the assignment we're working on today. The book, and the traditional old school name for these genes is they're called segment polarity genes. And the polarity is like a north-south sort of polarity. The polarity is, are you in the anterior team or the posterior team? And so it says here, segment polarity genes help to define the anterior and posterior polarities within each embryonic parasegment by regulating the transmission of signals via the Wnt signaling pathway and the hedgehog signaling pathway. So Wnt signaling pathway, we did that assignment. We're just going to recreate that assignment. Hedgehogs, we talked a little bit about that. It's going to be in here too. Okay, the book mentions this, textbook uses this word parasegment instead of saying compartment boundary. When you look at an insect, you say, I see its segments. Those must be really important in building the body. Segment, segment, segment. When you see us, you don't see those kind of segments, but you see vertebra, vertebra, vertebra. And we talked about somite, somite, somite. They're put together in a different way, but the whole idea is we're going to make populations of cells, and then we're going to have them on teams all the way down the body. So that's what's going on, just a different mechanism. The last common ancestor between us and arthropods didn't have a very segmented body plan, so those evolved independently, and they used different genes, like that, right? Yep, okay. So this parasegment bind is this invisible line. The book uses that. It's kind of old school. The reason it exists is because that's the organizing line that's set up. In the animal, we see the segments. The segments seem really obvious and important. but it's not the segments that matter. It's this invisible line down the middle of each segment. That's the organizing feature, like that, okay? All right, so the function of these segment polarity genes is to create an anterior compartment and a posterior compartment in each segment. So a population of cells that are going to be anterior team, they're holding hands with elbow macaroni, and a set of cells in the posterior compartment that are holding hands with shell macaroni noodles. Different cell adhesion molecules, the teams stay separated, they don't mix, and they create, as a result, an individual population, an individual line down the middle. They do this by creating this parasegment boundary, or more appropriately called these days, compartment boundary. Compartment boundary is a good term because this concept of compartment is used a lot. And you know the word compartment, like from a glove compartment. A glove compartment is just a special area where you can put stuff. And so a compartment is a story in embryology and developmental biology of, it's a population of cells that are on the same team, and they are on the same team because they're holding hands with these cell adhesion molecules, okay? So that's that. And then here's just some example of these segment polarity genes that we'll learn about. So one is sonic hedgehog. And we learned about that a little bit. There's a sonic hedgehog ortholog in us that patterns the different digits of our fingers. And it's used over and over and over as a morphogen in our development. It patterns the soma. It patterns our brain. It just patterns, patterns, patterns. So it turns out the original ortholog of sonic hedgehog was called hedgehog in fruit flies. We don't need to learn that name because the sonic hedgehog name is the much more common name now. What happened is when scientists found it in fruit flies, they called it hedgehog. And they called it hedgehog for a reason. So let's just look at that for a second. So here's a fruit fly life cycle. And so, you know, fruit flies are flying around. They develop initially as these little larvae. So the equivalent of a caterpillar in a fruit fly is a larva that's accurately called a maggot. So houseflies and fruit flies make maggot larvae, and they're really simple little gross worms. So it's going to grow like this, and that's what the geneticists were looking at when they were looking for mutant phenotypes. So the I story was looking at the adult. The early stories in the genetic screens were all looking at these little gross maggoty worms and seeing if they had a defect. And here's an example of a little maggoty thing, a fruit fly maggot, and it looks like in 3 different pictures. The easiest one to see is this one. So it's got segments, and on the posterior part of every segment, there's some little bristles. And it uses that for traction, like some people put studs in snow tires for traction. These bristles are just for traction, and it's going to squish its way through gross, rotting fruit. And so it has that, and they're on the posterior, and that allows it to sort of squish forward appropriately, where you have a little traction on the back edge of every segment, on the posterior part of every segment. So that's the wild type look. And so these people looked at those, and they found a mutant where the bristles were on both the posterior and the anterior. There was a mixing together of the 2 compartments so that they had these bristles, and the little things couldn't move around because they couldn't get the right kind of traction. And when they saw that, it looked like that, and they called the mutant gene hedgehog, because the mutant phenotype, they thought, looked like a hedgehog, a little curled up ball of bristles. Okay? When people found the ortholog in vertebrates, they were so ticked off already by fruit fly dumb names, they said, we can name it even dumber. We'll call it sonic hedgehog when you find it in a vertebrate. And you're just like, so that's the origin of that name. The name that sticks is sonic hedgehog because nobody cares about fruit flies and everybody cares about, you know, our body like that. So one of the genes we'll learn about is sonic hedgehog. Another one is wint, which was originally called wingless in fruit flies. Then we're going to learn, and these names are really sort of Drosophila specific, and so in Grail and CI and DPP. And we just talked about this right now. Sonic hedgehog, Wnt, and DPP are secreted signaling factors that go between cells. So they all have the same story. They're just kicking a message from one to the other. In our story, Wnt is going to be released by the posterior team and affect the anterior team. Sonic hedgehog is going to be secreted by the posterior team and affect the anterior team. They're going to be talking across this border using those two very, very famous signaling factors. The signaling factors have receptors. We know about Frizzle. Frizzle must be on the posterior compartment team's cell membrane, so they can receive the signal, and the signal is be a posterior team member. So a big part of our assignment is to make a list of some of the features of posterior compartment team members. Okay? Then, and GRAIL and CI are transcription factors. So they're going to be targets that are expressed differently. And GRAIL is going to be expressed and active in the posterior team, and CI is going to be active in the anterior team. Okay? Our book has this story, but I think our book jumbles it around. See, it's talking about the denticle, or I use the term denticles, the denticle belt, the bristles, and it says, look, you know, if you have a mutation, you're going to have a mess up in the phenotype of the little maggot. So our book goes through this, and it talks about this. This is just showing that wild-type animal has these compartments with this invisible compartment boundary down the middle of the wing, and a mutation could lead to a disruption in that distribution, and then as a result, it doesn't show that, but the wing would be defective. I guess it's showing there would be a mutant phenotype. There would be a defect if we don't keep that compartment boundary line going. It's an invisible line. You can't detect it without looking at the expression of proteins and seeing the proteins are different in the different compartment boundaries. Okay? So this is a similar story to what we already talked about. So remember when we talked about muscles at the start of our course, we were trying to emphasize that they're muscles because they express muscle genes. Our brain cells express brain genes. All the cells have all the genes, but they express different sets of genes, and that defines the cell. So we talked about that with muscles. You know what muscles are, and they're muscles because they express muscle genes, and we know how those get activated. So then we also talked some about stem cell identity genes. Those are the Yamanaka factors, and they're transcription factors. They're going to activate genes that are required to be a stem cell. Then we talked a little bit about brachyury. Brachyury is expressed in that belt of cells that are mesoderm cells. And those need to become mesoderm cells. Initially, they're specified as they think they're going to be ectoderm. And we're going to be like, no, we want you to be mesoderm. So we need you to express some mesoderm genes like brachyury and go down that pathway. So our new example is these segment polarity genes are doing that same story. They're all involved in expressing a certain set of genes in the posterior, including genes that make these cell adhesion molecules so the posterior cells will hold hands. And there's a different set of genes that are expressed in the anterior, and including a different set of cell adhesion molecules so the anterior cells will hold hands. And what we're going to do is we're going to just draw this out. We'll go on macaroni to underscore the significance of those, and then we'll especially pay attention to the posterior cells, and we'll make a list. And the list will be the Wnt is coming from the source cell, anterior compartment cells, and it is binding to the frizzled receptor on all the posterior cells, and it's the signal that's making them be posterior. So it's like we have two teams, and we start out together, mixed up. And then we're like, let's get this team over here. And then let's really establish who's on what team. And so all of the students on the anterior team start shouting, be a posterior team member. And the posterior team members receive that shout, which is Wnt, through the frizzled receptor, and they activate gene expression to become posterior cells and continue to be posterior cells forever. So it gets set up very early, And we're gonna have that line, and then the line will be the basis for further development, to build the different legs and segment body parts like wings and stuff in the arthropod. Yeah? Okay, so that's what we're up to. And this is a picture that is of what we're gonna draw and build in our assignment. So we're gonna jump into that. We won't do a concept chat today because we'll do this. And it's 2 cells. We're just gonna have a 2-cell drawing, but it's for the whole population. We can pick, Chloe as an anterior cell and Alex as a posterior cell to represent the whole team, because the whole team is doing the same thing, okay? So we're gonna make a drawing, and it's gonna be 2 cells, and the trick of it is that this will be a source cell making Wnt for that target cell. And then part of being a posterior cell is to activate expression of hedgehog and shout at the anterior cells, be anterior. And it's a feedback loop that's going constantly. That's what they're doing. They're shouting each other to be the other team. And that process just continues, and we get a really crisp boundary between these 2 populations. At first, they were a mixture. And the crisp boundary is based on the logic that sponges have of how they build their body. It's an ancient strategy to take a population and say, we gotta get some work done. Let's make 2 teams, and we'll get the work done between the 2 teams. We'll say that's our sight line, and we'll build stuff based on that line we established very early. And it continues for the whole life, right? First makes a maggot, later it makes a fruit fly, and it uses the same signals because they just keep going, okay? So let us do that. So if you got in here a little bit late, when if students were here, they already got some stuff, and the stuff is back there, and we're gonna start building. And like I said, you can sit together and build your own, but you can sit next to somebody and share stuff and cheat off each other, stuff like that. So if you want to make a group of 2 or 3 or 4 like that, that's great. We have our special guest, Sarah, so you can go sit next to Sarah. She's passing this class without taking any exams. How's that work? Yes. Just attending 1 class meeting and hoping to pass. What are the chances? Okay, so what do you need? Oh, Jesse's working on it too. What do you need, Jesse?
Is there a way we can pass without taking any exams?
Talk to Sarah. Sarah's got it. She's got a secret, go submarine route like that. Yep. Okay, so what you're gonna need is 2 pieces of paper, and you should tape them together. And I'm thinking you should tape them together like this.
Oh.
And you should use a piece of colored tape to tape the 2 pieces together. So that's what you need, just a strip of colored tape. And that's going to be the compartment boundary. So we're going to have 2 pieces of paper that are going to represent the anterior compartment and the posterior compartment. And then there's going to be this colorful tape between them. So you can put on a piece of colored tape. That's the only reason we need the tape, is to tape those 2 together. And you can label it with a marker. There are some of the markers are Sharpies. So they're the different shaped ones, and they're the best for labeling. And you could use a pen or something like that. But the other markers are a little bit, they kind of rub off when they dry. And I have a bunch of these markers. Some of them might be dried out. So if you run into a dried out marker, you can just put it up here and get a different one. And this is one way we'll identify which of the markers I should throw away. So if you find a dried out marker, just put it up here, the trash can, like that, okay? So now we have those, and what we should do is, and I'll draw it up here too, is on the top of the left piece of paper, you can just put a little label that says anterior compartment. Pretty small, but that's part of it. And then on the right piece of paper, you can put a label across the top that says posterior compartment, because that's what we're going to represent. Like that. And then, I'll just erase this so I can make it a little bigger. Just to give you a little outline vision of what the drawing's going to look like. This is just, now I've got up here, it's just one piece of the paper. And what we're gonna do is we're gonna make mostly just one big cell. And don't make it quite yet, because later, we're gonna wanna make just the edges of some of the other cells nearby, like that. But we just need to see a little edge of them. And these are the cell membranes, because that's where we're eventually gonna glue our macaroni. We're gonna glue macaroni around your big cell, and then we wanna show that it's touching some of the macaroni on the neighboring cells, because those are the handholds with the other cells. So that's just one of the pieces of paper. And the other piece of paper will be exactly the same type of thing. We'll have one big cell that is our focus, our example, our Chloe, and surrounded by just the edges of other cells so that we can add that glued on macaroni. And then in the other compartment, we'll have our Alex and one big cell of that, okay? So I'm gonna erase that just so I show the full view, but that's one each page, okay? So here's the two big cells. Like that, maybe I won't have enough room. And there's the anterior and the posterior cells that we're gonna talk about. And there's the compartment boundary to tape between these. Okay, so the first thing we're gonna add, and you know, we have a limited number of colors, but try to use a variety of colors. I didn't mention this, but what we're trying to do is make a memorable assignment. So someday when you're in the nursing home, somebody will say something about something that's on the news, and you'll be like, oh yeah, I think we use that as macaroni in college. You know, like a kind of unforgettable thing, because this is, you know, the compartment boundary, is it as significant as the hemoglobin in our body? No, it's crazily more significant for life on Earth than hemoglobin. Most animals don't have hemoglobin or insulin or bones, but every animal on our planet really has a lot of these compartment boundaries, and they set them up using different mechanisms, but they're trying to set up a crisp boundary. So what we want to draw here is our first thing, and that's going to be Wnt. Wnt is coming from the anterior compartment cell to the posterior, so make some little Wnt proteins like that. And you can just draw in here just a little bit, because we're going to add several genes, but here's just a little, you know, drawing of a little gene. That's the Wnt gene, and it must be getting expressed here. So part of being the anterior team is going to be to make Wnt. All right, so we'll get to that, but just leave space so we can put maybe 5 or 6 genes in each cell that are going to be expressed in that cell. And you don't need to write this down because it's going to get complicated, but right now, the anterior cells, those are source cells. They're making the signal. And then you can pick a different color, and then we want to put in this posterior some purple, and that's going to be the receptor for Wnt, that's frizzle. And the posterior cell is the target cell. And what we're doing right now is we're just recreating your assignment when we did a Wnt drawing assignment, remember? And there was a source cell and a target cell, and we drew that as general thing, okay? So it must have frizzle. So what do we know about posterior cells? And now we're gonna especially kind of organize the story in the posterior cells. So we wanna kind of organize this and say, here's frizzle gene, and then somewhere in here, try to make like a little, you know, like boxed in area in the posterior page where we're going to have a list. And the first on our list is going to be frizzle. And the list is going to be genes that are expressed in the posterior that are what we will call posterior identity genes. So to be a posterior cell team member, you need to express frizzled. So you can hear the anterior cells shout, be frizzled. And if you have room, you can just make a little parentheses, because you also need to express disheveled and GSK and beta-catenin. Because those are all proteins that are part of the Wnt signaling pathway, so we got to make them. And it would be good to pick a pretty color for beta-catenin, because that's the big show here. Because in the posterior cells, we've activated beta-catenin. and it can bind to the regulatory region of beta-catenin target genes and make sure they're going. So part of being posterior is becoming posterior, and that means to stabilize the beta-catenin. If we just glance to your left page, it has the beta-catenin gene and the frizzle gene and the GSK gene, right? They're genomic equivalents, but they're not being made. Beta-catenin is getting made but destroyed. So inactive in the anterior cells, active in the posterior cells. So then here's the next one on our list. We're going to talk about some targets in the posterior. So here's the next one on our list, and it's off of our important proteins that we already mentioned. It's called engrailed. So engrailed, it must be a gene, and it's getting expressed. So it's pretty safe to say that there would be some beta-catenin binding to the engrailed regulatory region, activating engrailed. And why is engrailed on our list? Engrailed in our story is comparable to the role that myogenin played in our muscle story. Engrailed is a transcription factor, and so it's going to help activate target genes that are posterior genes. And I'm sorry that it's hard, like I'm drawing little. But so if you're squinting like that, it's because I'm left-handed and a lousy drawer. But just ask, be like, what do you got up there now in that, red color, right? Because I'm trying to squeeze it in, and you're hopefully making it bigger. Maybe you'll bail and tape together 4 pieces of paper or 18 pieces of paper. Do it. And but we're making a list of genes that are going to be active in the posterior cell that are going to establish the identity. And what? Sorry?
What's the green?
The green is that beta-catenin, I'm saying. So we know about that. Beta-catenin is this transcription factor that gets stabilized, and it's going to bind the regulatory region of the gene and activate them. So it's going to probably help activate genes like engrailed. And the point of adding engrailed is that engrailed is going to help more. Its function is a transcription factor, so it will have additional target genes. Because of that, we can't say that for every posterior gene, it must be beta-catenin activating it, because it could be engrailed. So see, beta-catenin gets some helpers to activate all the necessary genes. The same was true, we didn't say it clearly then, but what's MyoD doing? It's activating myogenin in muscles to get some more helpers to activate more target genes, because we have several target genes. So part of being a posterior identity cell is to have the Wnt signaling pathway to receive the Wnt signal. Then the next thing is to activate engrailed to help turn on posterior genes. And I'm just going to erase mine and draw a little bit more. So then the third thing that we're going to say is this. Another posterior identity gene, so the third one on your list, is going to be what we're calling sonic hedgehog. Hedgehog in fruit flies, but we'll call it sonic hedgehog. And so there's a sonic hedgehog gene here, and maybe it has beta-catenin bound to it, or maybe it's engrailed. We don't know for sure, but it's one of those transcription factors that's activating sonic hedgehog gene. And sonic hedgehog protein, we know what happens with that. It gets made and gets secreted. So there's sonic hedgehog, it's a secreted signaling factor. And now your posterior cell is a source cell, and the target is the anterior cells. So you can draw on this, and we're not going to focus on this too much because we haven't studied sonic hedgehog much, but there must be a receptor here in the anterior cells, and it's actually called patched. So weirdo name, thank you for geneticist. But super famous receptor because it receives the sonic hedgehog signal, which is the signal that patterns our fingers and our toes and our somites and a lot of our nervous system. Our face gets zipped together with sonic hedgehog playing a really big role. And so patch plays a role like that, okay? And so we just want to emphasize, get a complete story on posterior, maybe not so complete. But let's make sure we do this, which is, as a target cell, the anterior cell team members, they must activate target genes. And here's one of them they activate. It's called Wnt. That was the start of our story. And we create this feedback loop where the anterior cells use Wnt to tell the posterior cells be posterior, and then part of being posterior is to use sonic hedgehog to tell the anterior cells to be anterior. So that's a key part of our drawing, and you might use some special color arrow to make a circular loop between that pathway, where Wnt is being expressed here to activate sonic hedgehog there, and sonic hedgehog is activated there to activate Wnt. And that's why we included beta-catenin, because that's a transcription factor that's the target of Wnt signaling. And that's why we just mentioned this CI up here, because CI is a transcription factor that targets by the sonic hedgehog. Yes, Al?
What are the blue and green dots?
So green is still, like, maybe it's beta-catenin,
Okay.
but, binding to the regulatory region of sonic hedgehog gene to activate it, or maybe it's in Grail. And it's true that, there would be some other transcription vectors that are playing a role too, but we just want to open our minds to the fact that it could be, could be one of a couple different transcription factors. We don't know which one. Scientists know, but we don't need to know which one it might be. It just shows that that's part of the story. We have these signaling factors, and their targets are transcription factors. And the target transcription factors are activating the identity genes. So sonic hedgehog is activating the identity genes in the anterior. Wnt is activating the identity genes in the posterior. So the way the cells establish the team are they're shouting at each other, be the other team. We hate you, be the other team, losers, like that. And they're shouting the opposite message like that to the other team, be posterior, right? It's a surprising analogy because usually when you have a team, you're like, yay, we're A team. But this is like, you guys be P. Yep, okay, all right. So again, we're not going to try to fill out everything in the anterior. We'll try to keep going with the posterior and get some ideas down like that, okay? So here's the next one, fourth gene that's part of being a posterior identity gene. And that is, and we're just going to use a general word. We're going to find out there are a variety of them, but it's going to be cell adhesion molecules. So I said those are proteins, and that means they must have genes, and we must activate some cell adhesion molecule genes by binding to the stabilized beta-catenin or binding to the engrailed that gets activated as a result of beta-catenin stabilization, and that these cell adhesion molecules get made. And now you get to use your macaroni, because you should show a posterior cell adhesion molecule as a particular type of macaroni, like either elbow macaroni or shell macaroni, and you should get some glue, and you should just put glue dots all around this cell and glue down pieces of one kind of macaroni in the posterior. And if you have some edges of other posterior cells, glue some macaroni there too. Because that's our new concept that we really want to remember here, is part of being posterior is indeed that feedback loop. That's what's establishing it. But then the functional significance of that is let's hold hands. So let's use a certain kind of macaroni cell adhesion molecule so we hold hands. Because that is going to create this crisp boundary where the two populations stay definitely apart. It's by the hand-holding cell adhesion molecule strategy. And you can add, and again, just add a bunch of macaroni that's the same kind to the posterior, and then you can do the same with the anterior, but use the opposite macaroni. So we have two different kinds of macaroni represented, elbow macaroni and shell macaroni. Because that's the visual we want to really get out of this. It's not just a colorful label, but like, okay, I see what's going on there. And if you have drawn the cell adhesion molecule, you might just be a little piece of macaroni, the appropriate kind of macaroni, right on the transcription unit, because that's that gene. And, you know, it's getting pretty crowded, but one thing to remember is the cells have all the genes. So let's say that you decided your posterior cell has elbow macaroni, and it's expressing elbow macaroni. Well, the anterior cells have the elbow macaroni gene, but they're just not making it. So the big story in identity of the cells is the gene expression. Muscle cells make muscle genes, and brain cells make brain genes. And of course, there are lots of genes that both cells need, DNA polymerase and so forth, but there are some tissue-specific genes that are only made in muscles, and some tissue-specific genes that are only made in brain, and some tissue-specific genes that are only made in liver. And this is like that here too. The way we can get the cells to become different is by having them express different genes. Yeah, they all have all the genes. We take any of these cells and turn it into a induced pluripotent stem cell to prove it has all the genes. But we want them to express some different sets of them in order to establish this boundary. Yep. And so we want to be able to do that, where we have a really good list focused on the posterior. There would be a comparable list in the anterior, which just would probably be, you know, start to be confusing. So let's just focus on the posterior and think about the list. So the first thing on the list is the posterior cells need to be able to receive Wnt signaling. So they need to make the Wnt signaling pathway. They need to make frizzled receptor. They need to make GSK. They need to make disheveled. They need to make everything to receive the Wnt signal, especially, of course, beta-catenin. They need to be making it because Wnt functions to stabilize beta-catenin. So that's the first example of posterior identity, is you need to be able to here be posterior and become posterior. And that's constantly happening as the embryo develops and the maggot crawls around and the fruit fly forms, because fruit flies have a very long development, right? They first make a maggot and then later they have to go through metamorphosis and make an adult. So this feedback loop just keeps going. So that's the first idea, is you need to be a posterior cell. You need to be hearing, I'm posterior, be posterior. Then, what's the next thing? Well, yeah, beta-catenin can help activate all the genes. Certain kind of gene it activates is like engrail. It's going to activate another transcription vector to help activate genes. So we're going to activate a bunch of genes. So that's the purpose of having that. There would be an example like that in the anterior, but we're just not going to go there. Then the third thing is, part of being posterior means to shout at the anterior cells, be the other team, be the A team. And that means you have to express hedgehog, sonic hedgehog. It's a gene, it's a protein, it's a gene, gotta express that. So one of these transcription vectors is gonna bind to it and activate that gene to be able to detect the expression of sonic hedgehog in the posterior, not the anterior. And then the next one is we need to express a distinctive handhold. So we need to express a cell adhesion molecule of macaroni type protein on the cell membrane that will allow our cells to hold hands. Yep, and when you get that all done, you should just take a picture. That would be the best way to get the assignment to me. And you know, sometime today, send me a snap of that. I'll have this, I have a tray, so if you don't want to take your assignment with you, you can leave it in this tray.
Yeah, you just went in
I
here
have this
also
tray
and
somewhere.
attached.
But
Just went and
I'm not
attached
really
them.
I don't really need to use your assignment to grade it if you send me a picture. So I'll have this up here, and if you want to just leave your assignment out here in the class, you can do that. Either way's fine, but just, you know, get it to me so you can get graded. Yep, And some people are using lots of macaroni, and that's awesome, because it's very pretty. And I apologize that I didn't like spray paint the macaroni, because that would be even more awesome. So do people have questions about that? I could have made this just a, it's another drawing assignment, but I tried to put macaroni on it for a couple of reasons. One, it's memorable, another, it's kind of big. And remember, I try to emphasize, make stuff big. Yeah?
Do have cell adhesion expression genes or just one?
They have all the genes.
Okay.
And there are many, many cell adhesion molecule genes. In chapter 7, where we really talk a lot about cell adhesion molecule genes, we'll find that humans have cell adhesion molecule gene families, right? So sets of paralogs that arose by DNA duplication, and they are maintained because they're of selective advantage. So primitive sponges just have a few types of cell adhesion molecules, both just a small number of genes and just one of each type. And then with more complicated bodies, we saw an increase in the types, but also in the families. So for some cell adhesion molecules in us, there are maybe 40 members of the family. And that just shows that it's just a profoundly significant tool for our development. Is that, I'm a mechanic, and I'm not satisfied with a toolkit that just has 2 or 3 screwdrivers. In my work, I need 40 screwdrivers, because I just have a lot of different stuff. A screwdriver, a cell adhesion molecule is a very valuable tool, and if I just have some different ones, I can make so many things. Remember, we started out saying you could take a sponge and you could separate into cells and just stir it up, and overnight it will just reform a sponge. In chapter 7, we'll see this. Remember, when the notochord forms in us and every vertebrate, it's going to induce the ectodermal cells to make our brain and spinal cord. Before the notochord is there, we have a population of ectodermal cells that's all equivalent. And the notochord is going to make them into 2 populations. The 2 populations of ectoderms are going to be ectoderms that are fated to form our skin, our epidermis, or our central nervous system. Really different fates, right? Different populations. In an early vertebrate, during that neurulation stage, when the neural tube is forming, as it's starting to fold up, you can take those cells, and you get the cells that are going to start becoming part of the neural tube, and you can take some cells that are not, that are in the ectoderm, and you can stir those cells together. And overnight, those cells will separate into the 2 populations. In these examples where we've seen cells starting to become different, very often, probably typically, part of the process includes expressing distinct cell adhesion molecules so they can hold hands and start being not just me. I can't really trust myself to do this job, but if I hear some support by holding hands with some other cells, I can start being convinced. And we'll see that in chapter 7, that when cells are holding hands, they actually receive signals from those hands saying, you're good, you're good, you're good to go. Some cells, if they're not holding hands, will just go through programmed cell death, because they'll just be like, I need a team. Yeah?
So the elbows hold hands with the elbows, and the shells hold hands with the shells.
Yeah.
Okay.
The concept.
Just wanted to make sure.
Yep. Everything you learn that matters, you learned in kindergarten. Like that. It's one of those kind of principles.
There's
Yeah.
a book that says
Very
that.
very physical story, right? They match up and they stay separate. And there's a mutation, it can give rise to a phenotype where those little bristles are scattered across the segment. And the little maggot can't get traction because it doesn't have the bristles limited to the posterior compartment. So it gives rise to mutations in these genes, give rise to very early and very bad phenotypes. Yep. Okay, so we're doing great, and we're set with that. Do people have questions about what we're working on? So let me show you that, go back to this, and show you that this is our first story in fruit fly. So on the second exam, or maybe the final, because that covers everything, you might have to draw this, or you might have to answer a short answer question, like it could be draw it all out, or it could be like to explain what genes need to be expressed in the posterior compartment of fruit flies in all arthropods. We're focusing on fruit flies. It hasn't been studied very much in other arthropods, but when it's studied, it's the same story. You know, a big old lobster has this story going on when it's a little embryo to organize its body into initially these parasegment lines, these compartment-boundary lines, and then let's build it. Oh, there's one more we want to add, and that is, so where we at like number, our cell adhesion molecules like our number 4 or 5?
4.
4. So let's add a 5, and day 5 is another gene that's on our list, and it's called decapentaplegic, but we're just going to call it DPP, and it's going to be expressed in the posterior, DPP. And we can just say this, DPP doesn't have an ortholog in us, but it's a member of this big gene family, TGF-beta. So we have a TGF-beta family member called DPP. And I'm going to just draw, and you can't get this on your figure, but it will help you understand how this system works. So if we look at segment, like on the edge, so let's just say that's the edge of a wing, just to see like something you can physically imagine. So this could be the compartment boundary. And we have the anterior and posterior compartment. And what we're talking about is we're saying that Wnt, so green will be Wnt, and this is not on your assignment, but just here. Wnt is going to be made in the anterior, and it's just going to diffuse a little way because it established very early that those are posterior cells. Very early, this was much smaller. Then, on the other hand, we have sonic hedgehog being expressed in the posterior. And these are paracrine factors, they just go a short distance. And so if we looked earlier in development, the wing would be very narrow, and you could see the job getting done in a very small thing. Now, how's it working when it gets big? And the answer is that these are referred to as short-range factors. And what we want to add here is DPP is a long-range signaling factor. So it is able to fuse much further, and it's a signaling factor. So it's going to affect target cells. And the source cell is these posterior compartment cells doing that. So that's the range of DPP, where it can spread out and really help pattern larger regions. And that should help you understand a little bit more about how we can use this signaling system that's just a line to create patterning across the whole segment. That's actually how the segment boundaries get established. So the segment boundaries you see are places where there's no DPP signal. And they're like, oh, I guess we should do a segment boundary because we're not near the compartment boundary. And so what we see in the animal later as the distinct body plan of segment, segment, segment is actually formed this way early. All right, so we gotta bail. We gotta get out of here. So if you don't want to carry your thing around and like immediately take it and show your friends and family, you can leave it here in this box. If you found some dry markers that are no good, you can put them in this smaller box. You can kind of put your stuff back. That would be helpful, you know, the markers and stuff. You don't really have to get them all sorted out because we got to get out of here. But yeah, we're good. So what's going to happen on Thursday is we'll get our exam back. We'll talk about that some, but we'll talk about the next of 3 stories. So that was our first story in fruit flies in chapter 2. And chapter 2 talks about it, it just adds more stuff and talks about some of the experiments and so forth that get done. Awesome.